So we're going to present what we did last summer, summer 2010, and we're doing this summer. Last summer, we were Biofilms, and this is just some background information. So we work with Saccharomyces cerevisiae, and short for like S. cerevisiae, as some people call it. Saccharomyces cerevisiae is a species of yeast cells. It's commonly known as like baker's yeast. People use it in like wine or like different things. And we also work with counter-alkins. The counter-alkins are also known as yeast, but they're a pathogenic fungus that binds itself to cell surfaces and causes like yeast infections, and it affects like 10 out of like 1,000 people a year. ALS5 is a mutant of Saccharomyces cerevisiae, and that's what we're using in our experiment this year and last year, and we used it last year also. ALS5 is also a gene of counter-alkins and is a protein of S. cerevisiae. Biofilms, which is the name of our experiment, are structures of community of microorganisms. And examples of biofilms are like soaps in your mouth, biofilm, and like moss, like when you touch like rocks underwater, like it's like the slimy effect. That's also considered a biofilm.
Last year we wanted to discover if ALS5 affects the way yeast grows, and we also wanted to know if ALS5 grows different from the other proteins, BADH, ALS1, ALS2.
These are our results for last year, and our results for last year we found out that the ALS5 does affect the way the yeast grows, because you can see on like these plates, which ALS5 is on the agar, the biofilms are larger, they're creamy, they're irregular. Well, some are irregular, some aren't. And these are the plates without the ALS5, so you can see that the cells are smaller. They took a longer period to grow to get to that size. So we know that when yeast is placed under CMS conditions, they do not grow as fast as when they're under the cells.
Okay, this year we have a different project. We are trying to figure out ways of how to promote and block biofilm formation. And we're using peptide A and vitamin T on CSM- plates. And the reason we're doing is that our co-mentor in the microbiology lab, she did an aggregation assay presentation, and she found out that when she used vitamin T with the ALS5 protein, that in high concentrations of vitamin T, that she found out that there was less aggregation of the cells. Aggregation means that there was less adhesion to really stick together. And so we are doing the same experiment, but on biofilms instead, to figure out if vitamin T and peptide A really affect the aggregation of the cells. Okay, amyloids are fibrous proteins, and amyloids is what build protein complexes. Each one of these are amyloids, and when they stick together, when they adhere together, they become protein complexes. So the vitamin T and with the vitamin T and peptide A, we want to figure out if they will go in between each of the amyloids, and so that they will form protein complexes. This just shows how the amyloids form complexes. Okay. So those each the amyloids.
They're attached to one another to form a complex. So the materials that we used this summer are plates, flask, incubator, autoclave. We had 3 different types of cells, which was ALS5 protein, the ALS5 E326N, and the empty vector. We use shaker, iPad, gloves, cistern thermometer, which are used to like just check the cell concentration of the cells that we're growing, Biofilms, which is gonna fluorescent, which is gonna show us the aggregation of cells and just adding. Well, at first we made 1 liter of CMS minus strip, and we made two 500 milliliters of CMS minus strip, but with 2 different types of concentrations of Biofilms, one 400 micromolar and the other 5 micromolar, because our co-mentor Melissa, who previously done a similar experiment to this, she found out that like if you use a high concentration of Biofilms, it's less aggregation, and if you use a low concentration of Biofilms, there's more aggregation. So that's why we used the 2 different concentrations. And then after we plated these on agar plates, we inoculated each of the strains on the plates, and we left them in the we inoculated them into like different suspension medias, and we let them grow in an incubator shaker. And then we took them out and tested the optical density, which just showed the cell concentration. And then after we got the desired results, we inoculated them all into different agar plates, and we placed them inside a 30-degree incubator. We observed their growth. So each week we're going to take out the plates and take pictures, and we're going to wash them out and see how they adhere to the agar. So every 24 hours for like 5 days, we're going to take out the set of counter outputs, and we're going to measure the plates in length and pictures and see the differences between all 3 of the strains, and we're just going to record our observations.
We didn't finish our project yet. The plates are actually growing right now, but our mentor did do a similar project, and when she did do it, then this was the first week of plates that she took out. And there's adhesion, but after you go to the second week, there's more adhesion. This is the adhesion of the cells. They're closer and they're bigger. So far, you see that after a couple weeks that there is adhesion. But this is only for CSM strip plates. We don't have the results of Biofilms and just that yet.
Right now we are waiting for the cells to grow, and we're going to observe the progression of the cells weekly, but we do expect that there will be more aggregation on the 5 micromolar than the 450 micromolar of the Biofilms concentration.
Our announcements will talk, you guys today, Dr. Peter, the CSM expert, our co-mentor, Chris O'Souls, the lab, Dr. Sad, and Dr. College.
Oh, 30 degrees Celsius, sorry.
That was the wrong side.
Why was that A?
No, again.
Oh, he asked why was the ALS5 red and the rest of them was white. Is that what you asked? In ALS5, when we made the suspension media, we had adenine, and adenine caused it to be red. And the rest of them didn't adenine.
So the ALS1 expressed on adenine.